Hello everybody, my name is Farhad. I would like to introduce you to MicroLoop. But what is MicroLoop? MicroLoop was designed for microbiome data analysis, especially for targeted or whole genome shotgun metagenomics data. It's quite useful for university students, for scientists, and also high-end decision-making bodies who are dealing with microbiome data. You can have access to it through Amazon Web Service, online, or if you would like to run it on your own computer locally, you can download it from my Docker Hub. So let's learn how to use MicroLoop. Imagine that you have a folder with your microbiome data, including a metadata, an OutU table, phylogenetic tree, representative sequences, and taxonomy classification table. Of course, you don't have to have all the data, but having a metadata, OutU table, and a taxonomy classification table is must. Your metadata should look like this, for example. You have a column with your ID for each sample, in this case, barcode number 1 to 36. You have your treatment, for example, other variables that are important in your study design. So this is completely study design dependent. Your OTU or ASV table should look like this. Column names should represent your samples, the sample IDs that are basically the row names of your metadata, and the row names should represent OTU, ASV, or MAG IDs. Your taxonomy classification table should look like this. Either parsed like this with 7 columns, each column represents one taxonomy level, or unparsed. And the row names here again represents the unique IDs to each OTU or ASVs. Once you have your data ready, you can go to the Count tab here and then load your data one by one. So let's begin with OTU or ASV table. Browse, OTU table, and then select the ID for that column that is going to be your row name, which in this case is ID. Then the data has header, yes. Metadata this is a CSV file, and like in this case, you can choose your delimiter. So it's quite robust in terms of recognizing different data with different extensions. Does your data have a header? Yes, it does. And I would like to give the name of that column that I'm going to use for my row names. And it comes to the taxonomy table. Our taxonomy data has a header, yes. And basically the column that I want to use as my row name is going to be that column that contains the ID of my ASVs or OTUs. And in this case, it's called ID. And my data is parsed. I can also load my representative sequences. Here, because I have my phylogenetic tree, I can also load it. It's quite optional. If you have it, it will be nice for phylogeny-based diversity analysis. And also your representative sequences, which is in FASTA format here. Once you have collected all your data, just click the button, Collect Data. And if you don't have them, you just want to play with the app and learn how to use it, you can click on Load Test Data. So here, let me introduce to you these different submenus. The first one is input data. The second one is for pre-processing your data. The third one is for alpha diversity. The fourth one is for beta diversity. The fifth one is for stacked bar plot, differential abundance analysis, association plots. If you have any numerical data and you want to create the association between those variables and your microbiome data, you can use this tab. And at the end, if you want to look at the phylogeny and different clades with different abundance of your taxa, you can basically go to the last tab. The first tab is input data, which basically shows you the metadata that have been imported, your OutU table, the distribution of the counts across all your samples, which in this case, of course, like most of the microbiome data, it's quite sparse, heavily tailed towards 0. You can always download these plots with different qualities, different dimensions, and also different formats. For example, if you want to download it as responsive, you can select the HTML format. If you want to download it as PDF, ready for publication, you can give it PDF with, let's say, 500 DPI. And then it will show it for you. We also have another plot that gives you an overall distribution of all the counts across the samples. This is the depth of your reads or the sequencing effort in each sample. Also, you can play with this slider to see how many samples will fall below a certain threshold of sequencing depth. Here we have the taxonomy table and also your representative sequences. And in each stage, when you have curated any data or when you created some new changes in your data set, needless to say, you can always download them here. Now let's go to the pre-processing by clicking on this red button. The first and foremost, this will tell you that your data, for example, in this case is 16S RNA amplicon sequencing data, contains bacteria in kingdom level, eukaryota, and unassigned. So the only one that I would like to keep in my data is bacteria because I'm looking at the prokaryota, 16S. There is another criterion for scratching out suspicious taxa in different taxonomy level, but by default it's on phylum, so just you could also stick to default. And here, if suspicion has been raised, this suspicious column will show you that that certain taxa, that certain phyla is basically erroneous and you can remove it from your data. Of course, you should know what you are doing. This app is offering you all these tools, but again, the decision is completely and entirely on you. In each step here, you can select whether to apply this step or not. Another one, you can see there is a table of suspicious taxa. If you select one sample to be the criterion. So let's say that how many samples should have a certain taxa so that that taxa is not considered error, but a biological variant. By default, I set it to 1 because I know the design of my study, but you can also play with it. And if this is a pilot study, or if the samples are coming from a certain rare and harsh condition, like for example, an environment like volcano, or the depth of the ocean, of course, you should be careful with this step, not to remove them, just keep them by selecting no. Now, another tuning parameter is for abundance-based filtering, basically, that finds the singletons in your data. And if you go down here and zoom completely in, you can find this is the distribution, the frequency of the counts of relative abundance without singletons. And at the top, it shows the singletons. So you can select yes, and then we apply pre-processing. And once it's done, you can again download this data because we have made some changes. Now that you are done, you are good to go to alpha diversity analysis. So here we are treating the data relative. If you have counted for the copy numbers, you can basically treat them as absolute count. Here, because we are looking at a diversity within samples, we need to basically take care of that differences, the variance that comes due to the sequencing machine and the human error. Therefore, we could do rarefaction, for example, that many people are doing. If you click on it, the rarefaction curve will appear here. Here you have the rarefaction curve with a default sequencing depth that you can play here and increase or decrease it. But let's stick to the default and see what 5,000 sequencing depth mean for this set of the samples. So each line represents one sample here, and you can also see them, select them or deselect them. And you can see that if I, for example, zoom a little bit in based on the richness, which is observed, if I select this depth, I will lose this sample, for example. And based on the Shannon diversity, you can see that I'm losing these 2 samples. I'm losing these 3 samples. So you can keep zooming in. The ones that don't reach the threshold line here, which is 5,000, they will be culled out from your data set. But we do rarefaction in order to unify the whole samples into one certain sequencing depth. Of course, in this case, since we have the selection without replacement, the likelihood of rare taxa to be selected is quite low. The odds are quite against them. Therefore, you need to be careful. Again, if you have sampled from harsh environments with a lot of rare taxa in them, you should not go for rarefaction. Instead, you can always go to cumulative sum of scaling transformation. And then you also have other options. You can play with them and see the before-after plot for the normalization. And then hit apply, skip, or revert. But in this case, I want to apply this transformation. And then you can see this is the plot the histogram of before and after. Now, I would like to visualize my alpha diversity. I have treatment, and I would like to have also faith phylogenetic diversity selected into my plot. I just hit visualize, and then you can see that I have a three-faceted plot. This is basically a box plot with a violin plot to show the density. So if you download this, plot is in front of the violin plot. You can also deselect them just to show the quantiles box plot. Or you can also bring more variables into consideration. And in this case, let's look at the weeks here. The second variable is weeks. So I'm going to bring weeks into my facet. And then I can see that week might have an effect on my alpha diversity. For example, you can see in week 0, my treated has already higher mean. By week 1, it's almost equal, and in week 2, it goes down. Therefore, in this case, I don't know if this is the treatment who's working or the week. Therefore, we call week a confounding factor. In this case, we can put the effect of week on the random effect by using a generalized linear mix effect model for alpha diversity. You can use LMER, GLMER, LM, or GLM. In this case, we stick to LMER and then fit the model. We put weeks as our random. In this case, the model is performing very well. Therefore, we accept that this model is fitting this data. This is for chow 1. This is for treated against non-treated. And you can see that it has a positive estimate, but the p-value is not small enough. And also our low confidence interval and high confidence interval, they contain 0. Therefore, we cannot accept that treatment had a significant effect on chow. The same scenario goes for Shannon and faith phylogeny. Now we are done with alpha diversity, and we go to beta diversity. Here, do you want to treat the data as raw or standardized from the previous? I also want to agglomerate my taxa to the highest taxonomy level, to the OTU or ASV level. In this case, it's species. I would like to stick to log transformation. That log transformation is performing great. Now I would like to visualize my raw counts based on a principal component analysis. In this case, this distance would be ignored. I choose treatment and hit proceed. If you see here, we have some clusters within different groups. These subclusters, we can find out where do they come from. We can, for example, define the shape based on the week. And there we can see that this subgroup belongs to week 0. Again, week is a confounding fact. I would like to choose distance-based redundancy analysis because in the design, I would be able to factor out the variance coming from weeks as a condition. Refit the model and visualize it. Now that we have separated the variance coming from week, you can be confident that treatment has a significant effect on this configuration of samples based on break dissimilarity distance. You can also choose other distances. Since we have our own phylogenetic tree, we can choose weighted unifrac. We can see that weighted unifrac also is confirming the same results. And here is your model extra. You can see in the conditional, which stands for weeks variable, it explains 12% of the variance. And treatment explains only 10%. And 78% of your variance was not explained. Therefore, this visualization is a magnification of this 10% that is explained by the treatment. And this is the results for your permutational analysis of variance, that again, with this small p-value, we can reject the null hypothesis that this configuration is by chance. But definitely treatment has an effect. Now that we are ready, we can take a look at the bar plots of all samples for all species or for all treatments and all weeks. And then see how it looks. This is how your stacked bar plot at family level should look like for this dataset. This is relative abundance, and these are different treatments in different facets of each week. Now that we are convinced that we had differences in treatment for beta diversity, we want to dig deeper by seeing that what kind of taxa have been differentially abundant to create this separation. Therefore, we do differential abundant analysis. Let's begin with species level. Here you have different modelers of your choice you can select. For example, we stick to the default DSEC2 and then create the study design. We are interested in seeing the effect of treatment on differentially abundant species. We will wait until the coefficients will appear here. Then we copy the coefficients and paste it here in the name field, and then select a certain alpha for our test statistics to be significant or not. And we can also decide on the color of my plot. Let's stick to the defaults. Here I'm gonna visualize a waterfall plot. Here we have log 2 full change, significantly log 2 full change based on the Benjamini-Hochbier adjusted p-value. And here is your table, you can also download them. And then on x-axis we have the species, and the color is for phylum. This is the waterfall plot of treated versus control. So if we have some species on the negative side, it means in the treated we have lower abundance of that certain taxa, certain species, compared to the control. If it is positive, it means it had higher abundance compared to control. We could also look at this plot in a different way, like a volcano plot. On the x-axis, we have negative log 10 for your p-value, which the higher, the more significant it is. And you can basically change that significance level here, which you can see this broken line would rise higher. And then here we have a corridor for the log 2 full change criteria. Here you can also download your tables. In some cases, you have numeric variables in your data and you want to see their associations between your microbiome data. For example, between different species, you want to check whether short-chain fatty acids, expression genes, body mass index, blood pressure, or diabetes index that have been detected automatically here as your numeric variables, whether they have any association between your different species. Here you can decide on the correlation method. In this case, I stick to the default Spearman. And then here is the number of taxa to be plotted in this association plot. And then here is the color palette for your association plot, which I'm gonna select red as my favorite color. And then here, since I'm deciding on visualizing the association between species and different numeric variables, I would like to also bring into account some lower taxonomy information. Like for example, I want to see the color of genus, orders, and different phyla. And then here you can decide whether the rows or the columns to be clustered or to be cut in different clusters. Then I decide to select this as yes, and also this one as yes. Then hit the create plot button. This is your association plot. So here we have the row classification, 1, 2, 3, as we have decided, and column 1, 2, 3. They have been cut based on their distance, WAR D2 distance. And here we have the information of your association. Lighter, more negative, more saturated, more positive association. And if the association in whatsoever case was significant, it would be labeled with a star sign. If we have our phylogenetic tree, we could also look at the phylogeny. We are looking at the data as relative abundance, and we would like to see the different species. And then I just hit create the tree to see how my tree looks like. And here you can decide on different taxonomy level. Let's stick to genus. It's easier to read, that's why. We can also select a taxonomy level for painting the branches. Now you can see that the branch color is phylum, and each clade that belongs to each phylum have been painted to the corresponding color. You can also decide on the shape of your tree. For example, we have fan as it is. If you want to have a better resolution of the differences between each clade, you can remove this. Here you are able to label some clades of your interest. I would like to realign them. You can also add tip points for better visualization, more appealing. You can also calculate high most common recent ancestors and visualize them, which in this case is family. You can also calculate the branch length and add it to the tree. Here you can decide which variable to be used to visualize the abundance around the tree. In this case, I would like to see the treatment for each treatment level, controlled and treated. And you can also decide whether this is relative abundance, log abundance, or absolute count abundance. And that's your phylogenetic tree. That's how you can have an in-depth overview of your microbiome data with high-quality, publication-ready visualization in no time, without knowing how to code in R or do some bioinformatics and statistical analysis.